The question of cell commitment and cell decisions is a huge question. These steps are lengthy. They take place during multiple stages of development across time. My lab set out to build the gene regulatory circuitry of a particular process in the cranial neural crest cells. It takes in account all the factors and predicts all their interactions and tell you who comes first, who comes second, who then activates the person that comes third, who can happen, who cannot happen. The whole idea of studying gene regulatory programs that define neural crest development stems from the desire to understand how these signals, factors, or downstream genes can go awry and cause craniofacial deformations, pigment cell defects, or heart deformations, for example. If we are able to define drivers that are affecting only a particular cell type, we are probably able to correct these in the future using new and coming technologies. The critical events in development of the embryos take place very, very early on. Perturbing instructions at the very beginning often will result in multiple outcomes. When we're talking about how many factors or genes are involved in this process, we're talking about hundreds and sometimes thousands. We joined forces with Fabian Theis's group in Munich. They are experts in defining developmental trajectories using RNA sequencing. It takes gene regulatory information and it takes trajectory information and makes its own network. So compute it together to give an idea of what's happening at the final stage if we perturb drivers or initial genes at the very beginning or along this way. Those two knowledges exist separately, but RegVelo is a model that integrates them and basically allows us to prove the validity of our discoveries. We're able to predict what are the essential drivers of particular cell fates and cell commitments, and then we're able to simulate this perturbation and then read out clearly the effect on very downstream outcomes. We were then able to test in vivo doing a large number of perturbations that were predicted to be significant. Why do we need to know? So you can imagine if you have an organoid, which is a very early set of cells, having a particular set of instructions could allow you to reproduce in vitro some of these cell types in a very natural way. And then these cells can be used in cell therapies in regenerative medicine.